Hello students, today we review on lipid metabolism. Mainly we will discuss about fatty acids, beta oxidation and ketone body metabolism, biosynthesis of saturated and monounsaturated fatty acids, biosynthesis of triacylglycerol and phospholipids or lecithins, biosynthesis of cholesterol according to the syllabus. Metabolism means change, a series of reactions. Any metabolism, that is anabolism and catabolism, are highly complex and continuous processes to keep us alive and involves thousands of different interconnected reactions to maintain the physiological steady state of our body as well as to maintain the homeostatic balance of our body. Metabolism of lipids in our body is also very essential for survival, growth and development. Lipolysis, oxidation of fatty acids are examples of lipid catabolism. Lipogenesis or biosynthesis of fatty acids, triacylglycerol, phospholipids and cholesterol are examples of lipid anabolism. Life could not exist without lipids. Lipid is very essential for all living organisms. Lipids are highly concentrated source of energy. Lipids present in our subcutaneous tissue acts as thermoinsulator and shock absorbing cushions. Phospholipids, cholesterol, sphingolipids, etc. are essential components of cell membrane. Bile salts, vitamin D, and different steroid hormones synthesized from cholesterol, prostaglandins, different eicosanoids, sphingolipids, glycolipids, and different metabolic intermediates of lipid metabolism have many biological functions in our body. In our body, lipids come from two different sources. One is exogenous source or dietary intake and another is endogenous source or endogenous biosynthesis. Dietary intake: except Rasgulla, some processed foods and soft drinks, most of the food contains lipids. Some food contains higher amount of lipids or visible fat, for example vegetable oils, butter, ghee, etc. But most of the food contains little or less amount of fat called invisible fat. Triacylglycerol, phospholipids, cholesterol ester, different fat-soluble vitamins, and many other types of lipids are present in food. When we consume these lipids in foods are digested and absorbed to the intestinal mucosal cell and forms chylomicron. Chylomicron transports lipid to the liver, adipose tissue, muscles and other organs from dietary source is called exogenous lipoprotein transport. VLDL from liver transports dietary lipids as well as endogenously synthesized lipids, mainly triacylglycerol, to muscle, adipose tissue, kidney and other tissues and forms LDL. This transport is called endogenous lipoprotein transport. LDL is mainly rich in cholesterol and transports mainly cholesterol to extrahepatic tissues and macrophages. HDL helps in reverse cholesterol transport, which we have discussed earlier. When lipids are transported by these lipoproteins to muscles and other extrahepatic tissues, they could not store. They utilize it except adipose tissue. Adipose tissue forms triacylglycerol from free fatty acids and glyceraldehyde 3-phosphate in the adipocytes for storage. In adipose tissue, 80% lipids in lipid droplets are triacylglycerols and are highly concentrated source of energy. Body do not prefer to utilize this storage when the blood glucose level is normal and also the amino acid pool is sufficient to produce energy. So when it is utilized, during starvation, prolonged exercise, pregnancy, stress, trauma, and other physiological as well as pathophysiological conditions when the blood glucose level goes down and blood insulin level is low. During hibernation, migration of animals, this stored lipid provides energy. But in humans, please see the 1 minute film. When stored lipid in adipose tissue undergoes lipolysis and after that produces energy by fatty acid oxidation during emergency for survival. One thing I want to mention here that during heavy exercise activity, muscles prefer to utilize glucose to produce energy more rapidly. So during starvation, migration, you have to work or move slowly to utilize fatty acids for ATP production by muscles, which will help to supply more amount of glucose and ketone bodies to the brain that time. Another important thing for oxidation of fatty acids is oxygen. Oxidation of fatty acid requires more oxygen than carbohydrates and proteins. Many times we have to face the question why fatty children feel hungry or cry for food. To get the answer, we have to start to discuss from the activation of hormone sensitive lipids, which is essential to start lipolysis and which is mainly controlled by endocrine system. And after lipolysis, the transportation of fatty acids from adipocytes to myocytes and hepatocytes take time because rate of diffusion of protein bound fatty acids are slower than free glucose and amino acids, and it enters into the mitochondria through a series of steps. And lastly, oxidation of fatty acid needs more oxygen. So, cells always prefer to utilize glucose and amino acids to produce energy. When there is no option and the cell is not in hypoxic condition, cellular glucose and ATP level is very low. Cells of cardiac muscles, skeletal muscles, smooth muscles, liver, renal cortex, brown adipose tissue, etc. start to oxidize fatty acids to produce acetyl-CoA. This acetyl-CoA undergo tricarboxylic acid cycle to produce NADH and FADH2, which by electron transport chain produces ATP. Acetyl coenzyme A in liver also can produce ketone body for gluconeogenesis to supply glucose in brain and RBC during that time. So now we discuss about the activation of hormone sensitive lipase to initiate lipolysis. When the blood glucose level goes down and blood insulin level becomes low and blood glucagon, adrenaline, noradrenaline, growth hormone level rises, they activate hormone-sensitive lipase in adipose tissue. Adrenaline and glucagon play important role in the hormone-sensitive lipase activation process. Adrenaline or glucagon bind with their respective receptors, which is intracellularly coupled with G-stimulatory protein, which activates adenyl cyclase. Adenyl cyclase converts ATP to cyclic AMP. The cyclic AMP level rises inside the cell, which activates protein kinase A. Protein kinase A phosphorylates perilipin and uncovers hormone-sensitive lipase for activation. Hormone-sensitive lipase has two forms, dephosphorylated form, which is inactive, and phosphorylated form, which is active. Insulin activates phosphorylase enzyme, which dephosphorylates hormone-sensitive lipase when the glucose level is normal. Glucagon or adrenaline, by activating protein kinase A, phosphorylates hormone-sensitive lipase. Thus, hormone-sensitive lipase become active during starvation, prolonged exercise, and migration. Hormone-sensitive lipase in adipose tissue acts only on triacylglycerol and produces free fatty acid and diacylglycerol. Then diacylglycerol lipase and monoacylglycerol lipase act subsequently to produce free fatty acid and glycerol. Adipose tissue could not catabolize glycerol because glycerol kinase enzyme is not present in adipose tissue. Adipocytes can only utilize glyceraldehyde 3-phosphate produced during glycolysis to form triacylglycerol. So glycerol comes out from adipocytes to blood by diffusion and transported to liver. In liver, glycerol is phosphorylated to glycerol 3-phosphate by glycerol kinase. This glycerol 3-phosphate is converted to dihydroxyacetone phosphate, which can undergo glycolysis, or it can be converted to glucose by gluconeogenesis. Free fatty acids bind with albumin in blood and transported to the liver, muscles, kidney, and many other tissues for metabolism to produce energy, except brain, because it could not cross the blood-brain barrier, and RBC also could not utilize fatty acids because it has no mitochondria. Fatty acids have to be activated to acyl coenzyme A and then transported to mitochondria prior to oxidation. In presence of ATP and coenzyme A, acyl CoA synthetase catalyzes the esterification of free fatty acids to fatty acyl CoA. This reaction is called activation of fatty acids. In this reaction, a thioester linkage is formed between the fatty acid carbon group and the thiol group of coenzyme A. The ATP undergoes pyrophosphate cleavage to yield AMP and inorganic pyrophosphate. Acyl-CoA synthetase is present in outer membrane of mitochondria, endoplasmic reticulum, and peroxisome. There are different types of acyl-CoA synthetase specific for their different fatty acids for different chain length. Acetyl-CoA synthetase activates acetic, propionic, and acrylic acids. Medium chain acyl-CoA synthetase activates 4 to 12 carbon fatty acids. Long chain acyl-CoA synthetase activates 12 to 24 carbon fatty acids. They can activate saturated, unsaturated, and hydroxy fatty acids also. After activation, short-chain fatty acids can diffuse freely inside the mitochondrial matrix, but long-chain fatty acids, that is about 14 to 20 carbon fatty acids, enter into the mitochondria by the acyl carnitine shuttle. Very long-chain fatty acids, that is 22, 20, 20 or 24 carbon fatty acids, are oxidized in peroxisomes. Long chain fatty acids, that is 14 to 20 carbon fatty acids, can cross the outer mitochondrial membrane through pore channels but could not cross the inner mitochondrial membrane. Scientists I.B. Fridge and others showed that the stimulation of fatty acid oxidation by carnitine is due to the action of an enzyme, carnitine acyltransferase 1, present in the outer mitochondrial membrane. Carnitine or beta-hydroxy gamma trimethyl ammonium butyrate forms oxygen ester linkage between its hydroxyl group and fatty acyl group of fatty acyl coenzyme A. Carnitine acylcarnitine translocase, present in the inner mitochondrial membrane, is an integral protein and antiprotein which transports acylcarnitine inside the inner mitochondrial matrix and transports out same mole of carnitine. Then acylcarnitine reacts with coenzyme A, another enzyme, acylcarnitine transferase 2, present on the inside of the inner membrane of mitochondria, catalyzes the reaction to reform acyl coenzyme A and carnitine within the inner mitochondrial matrix. Now fatty acid is available within the mitochondria for oxidation. Now how fatty acids are oxidized within the mitochondria? To understand the biological degradation of fatty acids, German biochemist F. Nook carried out an experiment. He used phenyl residue as a reporter group because it is not metabolized and excreted in urine. Phenyl substituted odd and even chain fatty acids were given in diet of the experimental animal, dog or rabbit. When rabbits or dog were fed an even carbon fatty acids of different chain length labeled with a phenyl group at the omega carbon, phenyl lactic acid was excreted in the urine. When rabbits were fed odd chain fatty acids with omega phenyl derivatives, benzoic acid was excreted. From this result, it was understood that fatty acids are oxidized stepwise at its beta carbon and releasing 2 carbon units acetic acid at each step. Fatty acyl-CoA within the inner mitochondrial matrix undergoes only 4 steps to produce 2 carbon unit acetyl-CoA: oxidation, hydration, in oxidation and thiolation. Each cycle of beta oxidation produces 2 carbon unit acetyl-CoA and the carbon chain length of the fatty acyl-CoA reduces by 2 units. Lastly, butynyl-CoA, a 4-carbon acyl-CoA, is produced by beta oxidation of even chain fatty acid and undergoes last cycle of beta oxidation. If it is a odd chain fatty acid, final product is propionyl-CoA and does not undergo beta oxidation, and it is converted to succinyl-CoA, a TCA cycle intermediate. Acyl-CoA within the inner mitochondrial matrix is oxidized by the enzyme adjacent to the respiratory chain in the inner mitochondrial membrane, collectively known as fatty acid oxidase. There are different dehydrogenases, hydrolases, and thiolases. In the first step, acyl-CoA dehydrogenase catalyzes enzymatic dehydrogenation at the alpha and beta carbon to form delta-2 trans-enoyl-CoA. Coenzyme for the dehydrogenase is a flavoprotein containing FAD as a prosthetic group. Hydrogens from alpha and beta carbons are transferred to FAD to yield the reduced form of FADH2, which denotes its electron to the respiratory chain via a second flavoprotein, electron transferring flavoprotein, which in turn passes the electron to the respiratory chain. Acyl-CoA dehydrogenase also possesses a chain length specificity. There are different long chain, medium chain, and short chain acyl-CoA dehydrogenase. A genetic deficiency of medium chain acyl-CoA dehydrogenase decreases the beta oxidation of medium chain fatty acids. Then medium chain fatty acids are oxidized by omega oxidation to form dicarboxylic acids, which are eliminated in large amounts in the urine, is known as dicarboxylic aciduria. Step 2. In the second step, delta-2 trans-enoyl-CoA is hydrated to form 3-hydroxyacyl-CoA by the enzyme enoyl-CoA hydratase. The addition of water across the delta-2 trans double bond is stereospecific and results in the formation of ill stereoisomer of 3-hydroxyacyl-CoA, which is absolutely specific for second dehydrogenation step. This reaction has no chain length specificity, no cis-trans specificity, but D isomers produced from the delta-2 cis or delta-3 trans unsaturated acyl-CoA could not undergo second dehydrogenation step. Step 3 or second dehydrogenation step. L3-hydroxyacyl-CoA is dehydrogenated to form 3-ketoacyl-CoA by 3-hydroxyacyl-CoA dehydrogenase, which is specific for A stereoisomer but has no chain length specificity. In this reaction, NAD+ is the coenzyme and acts as a specific electron acceptor. NADH is formed in this reaction, which donates its electron equivalents to NADH dehydrogenase of the mitochondrial electron transport chain to produce energy. Last step of beta oxidation is step 4, the thiolysis or the cleavage step. In this step, acetyl-CoA acetyltransferase, or more commonly known as thiolase or beta-ketothiolase, catalyzes the cleavage of 3-ketoacyl-CoA by the interaction with free coenzyme A to yield the carboxyl terminal 2-carbon unit as acetyl-CoA. The remaining fatty acids shorter by 2 carbon atoms, acyl-CoA is formed and enters the next beta oxidation cycle. The thiol group of the active site of the cysteine residue of the enzyme binds by thioester bonds to ketonyl oxygen of 3-ketoacyl-CoA. Then alpha and beta carbon bond is cleaved and acetyl-CoA carboanion and acetyl enzyme thioester is formed. Then acetyl-CoA carboanion is protonated to acetyl coenzyme A and a coenzyme A is transferred to acyl group to form acyl coenzyme A. Unsaturated fatty acids are mainly cis isomers in our body and are also oxidized by beta oxidation. Beta oxidation of unsaturated fatty acids are also continued within the mitochondria
until either a delta 3 cis acyl CoA or a delta 4 cis acyl CoA is formed, depending upon the position of the double bond. For example, if it is a oleic acid, after 3 cycles of beta oxidation, delta 3 cis enoyl CoA will be formed, which will be isomerized to delta 2 trans enoyl CoA by a cis trans isomerase and then again undergo beta oxidation. If it is a linoleic acid, after 4 cycles of beta oxidation, delta 4 cis enoyl CoA will be formed, which by acyl CoA dehydrogenase will be oxidized to delta 2 trans delta 4 cis dienoyl CoA and then will be reduced by dienoyl CoA reductase to delta 3 trans enoyl CoA and then will be isomerized to delta 2 trans enoyl CoA by delta 3 cis delta 2 trans enoyl CoA isomerase. This is a positional shift isomerism, and then delta 2 trans enoyl CoA again undergo beta oxidation. Hypoglycin, found in unripe ackee fruits, is an amino acid which is metabolized in our body by deamination and oxidative decarboxylation to methylene cyclopropyl acetyl CoA, which inhibits acyl CoA dehydrogenase during fatty acid oxidation, or it inactivates carnitine palmitoyl transferase 1 or thiolases during beta oxidation. This is characterized by gastrointestinal illness, vomiting, hypoglycemia, and CNS depression. Severe hypoglycemia and seizures ultimately leading to coma and death, mainly in case of malnourished children. Ackee tree mainly found in West Africa and is the national fruit of Jamaica. It is delicious and staple food of Jamaica. Unripe ackee fruit, when it is shelled, it contains hypoglycin and produces toxicity, which is commonly known as Jamaican vomiting sickness. Similar thing happened in India last year. About 100 children of Muzaffarpur, Bihar died by lychee fruit. Very long chain fatty acids, that is C20, C22, C24 fatty acids diffuses into the peroxisome and thioesterified by very long chain fatty acid thiokinase to very long chain acyl CoA, which is then oxidized by aerobic dehydrogenase acyl CoA oxidase to form delta 2 trans enoyl CoA. This step is different from mitochondrial beta oxidation. In this reaction, FAD is reduced to FADH2, which donates its hydrogen directly to molecular oxygen, forms FAD and H2O2. A catalase converts hydrogen peroxide to water and molecular oxygen. Then, like beta oxidation within the mitochondria, delta 2 trans enoyl CoA hydratase catalyzes the hydration to produce 3-hydroxyacyl CoA, which is again dehydrogenated by 3-hydroxyacyl CoA dehydrogenase to form 3-ketoacyl CoA. Here NAD plus is also act as electron acceptor and NADH is produced. Then thiolase cleaves the 3-ketoacyl CoA into acetyl CoA and a 2-carbon shorter acyl CoA is produced. Peroxisomal thiolase has substrate specificity longer than C8, so peroxisome could not metabolize octanyl CoA and is transferred to mitochondria for further beta oxidation. Zellweger syndrome is a rare congenital disorder, typically appears in newborn, described by Dr. Hans Zellweger in 1964. It is a peroxisomal disorder that occurs when peroxisomes are missing or do not function correctly. Accumulation of long-chain fatty acids, hydrogen peroxide in cells may cause the damage. Child with this syndrome have poor muscle tone, skeletal abnormalities, distinctive facial features, hearing loss, vision loss, etc. Beta oxidation is the main metabolic pathway for fatty acid oxidation, but in our body alpha and omega oxidation of fatty acids also take place. In brain, alpha hydroxylase catalyzes alpha oxidation of very long chain fatty acids for incorporation into cerebrosides and sulfatides. For example, lignoceric acid, C24 carbon fatty acid, by alpha oxidation forms 2-hydroxy-lignoceric acid or cerebronic acid, which is incorporated into cerebrosides called cerebrons. Some hydroxy fatty acids produced by alpha oxidation may be oxidatively decarboxylated to produce odd chain fatty acids for incorporation into brain sphingolipids. Phytanic acid produced by dietary phytol is oxidized by phytanate alpha oxidase and then by oxidative decarboxylation produces odd chain fatty acid pristanic acid, which undergoes beta oxidation and produces acetyl-CoA and propionyl-CoA. Genetic deficiency of phytanate alpha hydroxylase may cause accumulation of phytanates in tissues which produce different neurological symptoms. This disease is known as Repsum disease. Omega oxidation is a very minor pathway by the enzyme omega hydroxylase, a enzyme of microsomal cytochrome P450 oxygenase system which is present in smooth endoplasmic reticulum of liver. Omega hydroxylase hydroxylates omega methyl group of the fatty acid to hydroxymethyl group and after oxidation produces dicarboxylic acid which can undergo beta oxidation from the both end of the fatty acid CoA and produces acetyl-CoA as well as C6 and C8 dicarboxylic acids, adipic acid and suberik acid respectively. Inherited deficiency of mitochondrial mid-chain acyl-CoA dehydrogenase may cause accumulation of mid-chain fatty acids and are oxidized by omega oxidation and produces these dicarboxylic acids and are found in urine, which is known as dicarboxylic aciduria. If the chain length of a even carbon fatty acid is n, produce n by 2 number of acetyl-CoA from one molecule of fatty acid. For example, palmitic acid, a 16 carbon fatty acid, will produce 8 units of acetyl coenzyme A by 7 cycles of beta oxidation. If the fatty acid is odd chain fatty acid and chain length is O, then O-3 by 2 times beta oxidation cycle will be repeated to produce O-3 by 2 number of acetyl-CoA and 1 3 carbon unit propionyl-CoA. For example, heptadecanoyl-CoA, a 17 carbon fatty acid, will produce 7 units of acetyl-CoA and 1 propionyl-CoA by 7 cycles of beta oxidation. Propionyl-CoA does not undergo beta oxidation and is carboxylated by propionyl-CoA carboxylase to methylmalonyl-CoA, which is then epimerized to succinyl-CoA by methylmalonyl mutase. Succinyl-CoA is an intermediate of TCA cycle. Vitamin B12 acts as a cofactor of methylmalonyl-CoA mutase, so its deficiency in pernicious anemia patient excrete large amount of methylmalonic acid in urine. During beta oxidation within the mitochondria of hepatocytes, acetyl-CoA is produced, which condenses with oxaloacetic acid to enter the TCA cycle. Excess acetyl-CoA within the mitochondria inhibits pyruvate dehydrogenase and activates pyruvate kinase to produce oxaloacetic acid, but oxaloacetic acid is utilized for gluconeogenesis and its level is reduced during starvation, migration, hibernation, diabetes mellitus, and other conditions when the blood glucose level is low or the glucose supply within the cell is restricted. During that condition, acetyl-CoA cannot be properly metabolized through TCA cycle in the liver and are accumulated inside the mitochondria. So thiolase or acetyl-CoA acetyl transferase present in hepatic mitochondria catalyzes the condensation of two molecules of acetyl-CoA to acetoacetyl-CoA. Acetoacetyl-CoA is also produced in beta oxidation during penultimate cycle. Some amino acids breakdown also produces acetoacetyl-CoA. For example, leucine, lysine, phenylalanine, tyrosine, tryptophan, etc., which are known as ketogenic amino acids. Thus, acetoacetyl-CoA accumulates within the hepatic mitochondria. Acetoacetyl-CoA again undergoes condensation with another molecule of acetyl-CoA to form 3-hydroxy-3-methyl-glutaryl-CoA or HMG-CoA by the enzyme HMG-CoA synthase. The enzyme HMG-CoA synthase is only present in hepatic mitochondria and is the rate-limiting enzyme for the formation of ketone bodies. Then HMG-CoA lyase present in the hepatic mitochondria catalyzes the splitting of HMG-CoA to acetyl-CoA and acetoacetate. This acetoacetate can be reduced within the mitochondria to 3-hydroxybutyrate by the enzyme 3-hydroxybutyrate dehydrogenase, or it can be spontaneously decarboxylated to acetone. Acetoacetate, 3-hydroxy or beta-hydroxybutyrate and acetone produced during high rate of fatty acid oxidation are collectively known as ketone bodies and are used as energy packets during glucose deficiency in body. Ketones and butyrate or these ketone bodies are soluble in water. This is the main specialty of ketone bodies and is different from fatty acids with long hydrophobic chain. Ketone bodies are also membrane permeable and can cross the cell membrane without any transporter by simple diffusion. Ketone bodies need no carrier molecule to transport within the bodies. So, ketone bodies from the mitochondria of hepatic cells come out and distributed by systemic circulation to most of the tissues and organs of our body. It can also cross the blood-brain barrier. Acetone is removed as gas from lung by breathing. 3-hydroxybutyrate or beta-hydroxybutyrate is more reduced form of ketone body and can release more energy than acetoacetate. And this 3-hydroxybutyrate is reoxidized to acetoacetate by 3-hydroxybutyrate dehydrogenase. In this reaction, NAD is reduced to NADH plus H plus. Acetoacetate in extrahepatic tissues thioesterified to acetoacetyl-CoA. This reaction can be catalyzed by succinyl-CoA acetoacetate CoA transferase or 3-ketoacyl transferase or thiophorase. This enzyme is present in extrahepatic tissues but not in liver, so liver could not metabolize ketone bodies. This enzyme catalyzes the transfer of coenzyme A from succinyl-CoA to acetoacetate. With the expenditure of ATP, acetoacetyl-CoA synthase can also transfer coenzyme A to acetoacetate to form acetoacetyl-CoA, but this reaction is less important. These acetoacetyl-CoA produced within the extrahepatic tissues are thiolysed to two molecules of acetyl-CoA and enter into the TCA cycle for energy production, or it can be utilized for gluconeogenesis also. Formation of ketone body within the liver is called ketogenesis. Breakdown of ketone body in extrahepatic tissues to produce energy is called ketolysis. Presence of ketone bodies in blood and urine is known as ketonemia. Excess ketone body within the blood is known as hyperketonemia. When ketone bodies are excreted in urine is called ketonuria, and which also causes ketoacidosis. The overall condition is called ketosis. Ketone bodies are rich energy sources and essential in some conditions in our body were discussed. But ketone bodies are also toxic and can cause mild or moderate toxicities in our body during prolonged starvation, severe exercise in post-absorptive phase, high-fat diet for long time, diabetes mellitus, etc. Now we will discuss about biosynthesis of fatty acids, triacylglycerols, phospholipids and cholesterol after a short break. Please open the textbook available to you and read thoroughly about the oxidations of fatty acids and ketone body metabolism discussion.